All right, everyone, welcome back. Now we're going to have the lightning talks portion of the agenda, where we're going to hear from 4 faculty on their research programs. And I'm just going to introduce all 4 at the start, and then they'll hand it from speaker to speaker. So first, we're going to have Jenny Kao-Kniffin, who's professor in horticulture and also the associate director of Cornell Cooperative Extension. And her lab has been doing some exciting work on soil microbiology and urban soils. Second, we're going to hear from Greg Peck, who's also professor in horticulture, who studies the sustainable management of perennial tree fruit and has been a co-lead on a lot of the great soil health work in orchards across New York State. Third, we're going to hear from Virginia Moore, who's a new faculty in plant breeding, who's been doing some really great work on breeding better winter annual legume cover crops and other areas. And finally, we're going to be hearing from Louis Longchamps, who's another new faculty in digital agronomy, who's working on building an exciting program to facilitate on-farm precision agriculture research across New York State. So with that, I'll hand it over to Jenny.
Thank you, Joseph, for organizing this excellent statewide soil health summit. This is very exciting to hear all these presentations. I was asked to give a talk about a project that I have to mention that my background is not urban agriculture. So the way I got into urban agriculture was a result of an initiative led by Max Pfeffer, who was our previous executive dean for the College of Agriculture and Life Sciences. Under the leadership of Catherine Bohr, who was our previous dean for the college. And what they wanted to do was to encourage more research and outreach in New York City, a CALS initiative in New York City. And so that particular year, this was back in 2019, they asked faculty to provide proposals on what they would like to study. And so I, along with Kyle Wickings is a professor in entomology, I'm in horticulture, we both decided to come together and prepare a proposal looking at green spaces. So basically turf grass. So that was our background. Turf grass in New York City, so parks and other areas to assess soil health in there and soil quality. And with me on the focus of soil microorganisms and Kyle with a focus on soil arthropods. Now, we were asked by Max Pfeffer to come together with another group that also wanted to work on soil quality. And that was a group led by Jonathan Russell-Anelli, who is one of our national experts on urban soils. So his team and our team came together and met in the middle and into this emerging area of commercial agriculture in New York City, focusing on soil-based systems. And so I'm just going to share a very short glimpse of this journey together that we've had since 2019. And I'll start with saying, can I advance my slide myself? Looks like, yeah, I can, thank you. And so I want to acknowledge the work of the Harvest New York Extension Associates. These are Extension Associates that are part of the Harvest New York area teams. So it's a statewide group at Cornell University and Cornell Cooperative Extension. Sam Anderson and Yolanda Gonzalez, they do the day-to-day relational work of training and education with urban growers in New York City across all 5 boroughs. This is their map. It shows you the footprint of who they work with, the very strong relationships they're building, and the very dynamic initiatives they have that include a large component on soil, on compost, because the important work by Jonathan Russell-Anelli, Murray McBride, Jean Bonatel, and Hannah Shaler show that what is really important over the past 20 years, the past 2 decades, is that using the endemic soils in New York City, it's very contaminated, so it's not very safe to use. But being able to cap that or provide some type of barrier and use existing soil, municipal compost, or exported or imported from outside New York City, that ensuring that they are safe and not contaminated with lead or other types of historical legacies of urban contaminated soils. So because of that, it's really been growing a lot of this urban growing of food, vegetables, fruits, primarily across New York City and other cities across the nation. And so here, our goal to work with Jonathan Russell-Anelli's team Kyle Wickings and I and Jonathan's team, what we decided to do was to work on some of these sites that Yolanda Gonzalez and also Sam Anderson work on. And it was just basically 33 sites that we wanted to look at. Here you see over 65 sites, but we wanted to do, less than half of that. And our goal was to do that starting in 2020. And if you can remember 2020, I will remind you in the next photo. So one of the questions that I'm sharing with you is a small snippet of the story we want to share. Do microbial communities converge over time to resemble similar taxa and functions? Microbial communities are a very important part of the aggregate soil biological component of soil. So soils are alive, and there are organisms that help it be alive, and they perform very important functions, such as nutrient cycling, the ability to promote soil moisture. They can produce exopolymeric substances that help create moisture zones in the soil. They can also produce glycoproteins that help create a sticky substance that play an important part in carbon sequestration and soil aggregation. I can go over an hour of this easily, but this is just 7 minutes. So I'll quickly go over just a small snippet of data. So we had this grand plan to do 33 sites, 2020 restrictions for those of you that lived in New York City and experienced, unfortunately, what happened. Our research team was bare bones. I had to bring my own family as my research crew, and we had to be able to synchronize teams separately in different locations. We could not stay in the same hotel rooms, cannot stay in the same vehicle. We had all these restrictions to keep everyone safe. So 33 sites ended up being 9 locations, but they were still across 4 boroughs, and they span from 1 year to over 25 years old in terms of the farm site and soils. And here I want to show you that I'm just focusing on soil microbiome data, and Harold Van Es talked about the amplicon sequencing. And this is an important part of microbiome analysis. What it allows you to do is look at a high resolution profile. You're able to extract DNA from the soils, and you're shearing off the cells of bacteria and fungi, getting their DNA as it's shown in the slide on the right. And you're using primer sets, this is molecular biology tools, to get at that specific gene. So for bacteria, it would be the 16S ribosomal RNA. And for fungi, it's the internal transcribed spacer region. And that gives you very detailed resolution of taxonomy and systematics of what you're studying in the soil. The next slide shows you some of the results. Now, this is very exciting to see because we really didn't expect that the bacteria would change over time. So if you look from right to left on my screen, so the lighter ones are the younger farm sites, and the ones on the left are the older farm sites. So as you get more red, those are the older sites. These sites are not next to each other. They're in 3 different boroughs, the older ones, and the ones that are younger, they're across 3 different boroughs as well. So what we find is that a lot of these sites that are focused heavily on inputs, so they're not endemic soils, these are inputs. So they're getting compost or getting soil mixtures. Some of these locations are getting soils and compost from Long Island Compost, which is now owned by Miracle-Gro, Scott's Miracle-Gro. Or I've seen locations with pallets of Scott's Miracle-Gro or pallets of sphagnum moss. So they're getting it from different locations, or they can get it from municipal composting facilities like DSNY and some of the other contracted locations. So this is a very fascinating result. What do the fungi do? They show a very similar trend as well. Again, the younger sites tend to be very similar in terms of the types of fungi. The ones in orange you can see in the top are the medium-aged farm site soils, and the ones on the left, they are the redder colored ones. Those are the oldest sites, the 22 to 25 years old. And these are different boroughs. And what we find is that for some reason, the fungi over time start to look like each other. And the ones that are 1 to 2 to 3 years old, that's an average, they tend to look similar. But over time, there's a convergence that these different locations, they start to have the same types of taxa, fungi and bacteria. And what we also see is organic matter declines over time are very clear, which is fascinating because these older sites are getting routine shipments of new compost and soil mixtures. It's not just, they had it once 25 years ago or 22 years ago, they're routinely adding this. In spite of that, the organic matter percentage levels are quite low at some of these locations. And we're doing a lot more analysis on the microbiome and assessing environmental variables to assess, what are some of these patterns? Are there specific types of microorganisms that are associated with low organic matter? Is these carbon dioxide soil respiration, is that declining over time? So is it greater carbon sequestration occurring in terms of recalcitrance? The next is takeaways. Do microbial communities converge on functions over time? What we'd like to know is some of their functions. So doing some substrate utilization profiling of carbon compounds, different types of carbon compounds and their utility of it, and types of enzymes that they're using to modify their environment. And so this just shows you photos of the 3 oldest sites and that converged over time. The bacteria and fungi tend to look similar, and there are 3 different burrows. The one on the left is Sam and Yolanda. Those are extension associates in Harvest, New York. And they're at Randall's Island, which is technically part of Manhattan. And on the top is Brooklyn, so East New York Farms, the 22-year-old site. And in the bottom, it's the oldest site in Queens Botanical Garden, managed by Karen Guzman. And finally, I just want to thank everybody. There are a lot of team members that are part of this and farm participants, a lot of farms involved as well. And that's all I have. Thank you.
Awesome. Thank you, Jenny. Next, Greg Peck.
Great. So I've got my soil health hat on now. I'm Greg Peck, and I'm an associate professor of horticulture, and my research program covers a lot of different areas. I did a lot of work in my early part of my career on organic systems, and that easily ran into understanding more about soil health, soil function in orchards. And those of you who might know me, also I work a lot on hard cider production as a whole other research area, and sometimes these 2 topical areas merge. But when we talk about soil health for apple orchards, there are some really key considerations, some things that are different than, in particular, how the soil health testing system has developed over time here at Cornell, but also some things that we need to think about differently because these are perennial systems and they're long-lived. They typically have deeper root systems, but not always. And as we start to see an evolution of our orchard systems from what we see in the top right-hand side here, these big trees, deep systems, root systems that can really explore the soil, to what we see on the bottom picture there with one of my former grad students, is that the roots aren't exploring as much of the soil volume. And so we really need to think about what's happening in that space and how, for what a lot of our larger scale commercial growers want, which are high density, very productive systems, how do we manage soil health? And it's often in terms of nutrient and water availability. So I think a lot of producers are, and this is true for both of our organic growers and our conventional growers, are really thinking about sustainability. They're thinking about how do we minimize fertilizer inputs. They're also thinking about it from the cost perspective. And we've seen some real spikes in nitrogen fertilizer costs over the last decade or so, often related to global political situation. So, click this button here. There we go. So been working with the soil health team for many years now. And in our last report from 2019, it was noted that our soil health policy research outreach efforts need to be expanded to, and I'll just highlight, apple growers. And again, this is because a lot of our previous work and a lot of the systems that were developed were for annual systems. So over the next couple minutes here, I'm just going to give you some highlights of some of the work that we're doing and some of the things I want you to think about. One area that we've had a lot of focus on for research is how do we incorporate more organic matter into orchard systems? So because these are perennial systems, we don't have the same ability to go and do a cover crop, to do rotational systems. We do this maybe once every 20 to 25 years as orchards get replaced, but not on an annual basis. So the way to add organic matter, one of the ways is through mulches. We also can add things like compost and other organic amendments, but mulches is one really interesting and I think potentially useful from a policy perspective as well, way of trying to increase organic matter, trying to increase soil health for orchard systems. And so this is work that goes back a long time to my PhD advisor, Ian Merwin, but we've seen research on this topic in Washington State, through the Midwest. Here in New York, Debbie, who spoke earlier, has done some of this work in Long Island. And certainly we've done a lot of this in upstate New York in the Ithaca area and also Lake Ontario. So by adding organic matter to the soil, adding mulches to the soil, we can have all of these positive benefits, increasing water holding capacity, increasing soil biological activity. We're increasing our mineral availability, suppress diseases, reduce erosion, possibly have some disease or weed suppression capacity as well, and reduce nutrient leaching. Another thing, and I'm really glad that Jenny gave the talk right before mine, is that you can also think about when we add organic matter to the soil, we're also changing the amount of biology that's in there. A lot of the microbial biomass increases, but also very specific things change as we add mulches to soils. So this figure here is from a paper we published a couple of years ago. And we see here, this is a fungal group of arbuscular mycorrhizae. And so this is a very beneficial group of organisms to increase in the soil. And you can see here on the left-hand side, these 4 treatments are all the mulch treatments. On the right-hand side are all the no mulch treatments. And you can see that the mulch treatments are increasing the mycorrhizal populations in the soil. We did some work looking at how mulches can help to suppress weeds, specifically in organic apple orchards. This is work by Kate Brown, a master's student at my program, just graduated this past year. And we were comparing a lot of different ways. Organic growers often point that weed control is their number one most difficult management thing to deal with from a management perspective, and especially in our humid climate with frequent rainfall, that weeds can really be a problem. So we're looking at how do we do this without the use of synthetic herbicides. And so one way is through mulching, and we found that it was beneficial that for soil quality, but that we found that cultivation actually resulted in the largest trees. So it's always a mix, and it's always trying to figure out the right strategy for the right place in the right situation. I want to go back to the microbial populations too, and this is a little different study, and we're looking at different compost amendments. And we can see really strongly here on the right, the green and the red colored ovals there. Those are microbial communities that are more closely related to each other, and that's for our control and calcium nitrate. But when we add either the purple, which is a yard waste compost, or the gray, which is a chicken litter compost, we can really change the microbial communities. And it's related to all these functions here: microbial respiration, nitrogen mineralization, and so on. So back to the big picture in the last couple of minutes here, and working really closely with the soil health team, we're trying to develop orchard-specific soil health measures. And this is all these dots here. Debbie put this map together. Are sites that we have been collecting orchard soils from, and we're trying to relate that to productivity so that we can develop new soil health tests that are specific for orchards. And there's others working on various aspects of soil health. I've mentioned a few people here. I want to mention some of our extension colleagues, Mike Bazow, Janet Van Zoren, who are looking at soil health and herbicides and mycorrhizal products. And we also know that we have a very strong program for rootstock breeding in New York with the USDA. And we're looking at the impacts of soil health on those. So where do we go for this from this point on is developing orchard-specific recommendations for soil health. Really trying to go further with this idea of exploring the microbial contributions to soil health and how that affects orchard productivity and ecosystem services. The third point here I really want to highlight right now too is to incentivize growers to improve soil health. And so this is something that is really important to think about from an orchard perspective. A lot of our work and others have shown that we don't always have an increase in yields, even though we have an increase in soil health. So we know that there's a positive benefit to it, and we may have to find other ways to incentivize growers to use these practices in their orchards. And we hope to also expand beyond orchards into other perennial fruit crop systems. There's been work in vineyards and berries. So thank you to all the people on this slide. And I also want to acknowledge and thank everybody who's mentioned climate change. This is a picture from one of our research orchards here in Ithaca from just a few years ago. And there were literally ducks swimming in our orchard because of the intense and repeated precipitation that we had. And that is just something that we're all going to be contending with and dealing with no matter what cropping system you work with. Thanks.
Awesome. Thank you, Greg Peck. And now over to Virginia.
Hi there. Yes, so today I'll be talking about opportunities for plant breeding as a tool to promote soil health. So I'm an assistant professor in plant breeding and genetics here at Cornell, and I also have a background in agroecology. And a big part of my role here is looking for ways to use breeding to improve sustainability in our agricultural systems. And so these are soil health and plant breeding are two areas of research that aren't always paired together, but I see a lot of opportunities to use ideas from both of these areas to enhance soil health outcomes. And so I'll just talk briefly about some of these the practices that other folks have been talking about today and how plant breeding might intersect with them. So if we think about maximizing living cover as a management strategy for soil health, we can think about breeding improved cover crop varieties or breeding perennial cash crops to improve their compatibility in our rotations. And we can maximize crop diversity as a management tool, right? And there's ways to do this to improve our ability to do this with breeding, thinking about breeding for intercropping systems, and breeding new cash crops or cash crops that are new to our region, right? Increasing diversity in our rotations. We can also think about using breeding to minimize tillage. And so this might look like breeding our cash crops or our cover crops for improved performance in no-till systems. And so I'll spend the rest of my presentation focused on the types of projects that my lab is currently working on that address these various areas and intersections between breeding and soil health. So the first area that I'll talk about is maximizing living cover through cover crop breeding. And so my lab is focused on several different cover crop species and a wide range of characteristics for each of these species. And in some cases, we are breeding directly for traits related to soil health. And we certainly are interested in doing more of this, thinking about root traits and symbiosis with microbes and improving cover crop ability to sequester carbon. But a lot of what we're doing is really about improving farmers' ability to adopt cover crops as a soil health practice. So we're trying to reduce the barriers that farmers talk about that reduce their ability to use cover crops, things like timing and seed cost and weediness that make it difficult to use cover crops on farms. And this is work that's being led out of my lab at Cornell, but really involves a huge range of collaborators. And you can learn more about our cover crop breeding network if you follow the QR code in the upper right corner of this slide. But I'll give a kind of a brief overview of that network. So it's a group of researchers at universities and at USDA ARS centers and collaborators at NRCS, some farmers that are doing on-farm breeding and variety testing, and extensionists as well that are helping promote appropriate variety selection for different regions. And this is a group that came together starting in 2015 and has been funded primarily by the USDA Organic Research and Extension Initiative, OREI. And I'll touch briefly on some of the projects that we're working on in the network. So first, winter survival in our region can be a barrier for fall planted cover crops, especially for legumes. And so one of the projects we're working on in my lab is identifying winter pea varieties and regionally appropriate planting dates that maximize the chances of winter survival and good biomass production in the spring. So this is work led by my graduate student, Raksha Thapa, and it's also in collaboration with some folks in Minnesota and North Dakota who've planted a replication of this experiment as well. And we're in our second year of this experiment with some promising results. Next experiment I'll talk about is similar approaches used in cereal rye. So this is an experiment evaluating just in New York State locations, looking at a range of planting dates and varieties in collaboration with some researchers and farmers in the northern part of the state. We've got 4 planting dates between September and early November, and we're looking to identify varieties that are better adapted to later planting dates so that farmers can more easily get cover crops in the ground after their cash crop is harvested. And we're also breeding rye with the goal of improving adaptation for northern environments. Looking at things like tolerance for late planting, as well as other common cover crop traits like early vigor and biomass and early maturity. We're also interested in allelopathy as a tool to improve weed management in cover crop production systems. Towards the goal of maximizing living cover, my lab also does a lot of breeding in perennial forage crops and variety testing under the leadership of Julie Hansen. And we know that forages and other perennial crops play a really critical role in our cropping systems and provide soil health and benefits and other ecosystem services. In terms of this goal of maximizing crop diversity for soil health purposes, we can think about breeding for diversity over time by, for example, diversifying our crop rotations. In our lab, we're working on developing hemp as a crop for New York State. This is in collaboration with Larry Smart's lab, as well as a number of other researchers at Cornell. We're really interested, especially in developing hemp within the context of sustainable cropping systems, thinking about integrating cover crops and reducing tillage as we develop this new crop for our state. And towards this goal, we're working on assessing breeding and research needs for organic hemp production system. Over the winter, we're going to be conducting some surveys and focus groups with farmers, industry folks, extension, educators, teaching about hemp. And if you're interested in participating, please reach out and keep your eye out for those opportunities. We also are thinking about maximizing crop diversity spatially. So looking at intercropping and mixtures of different crops. This could be row intercropping or strip intercropping, as well as crop mixtures. One place that we're doing breeding for intercropping is in alfalfa and perennial grain systems. So we're evaluating opportunities to intercrop intermediate wheatgrass or kernza with alfalfa to promote these dual purpose forage and perennial grain systems. And my graduate student, Megan Williams, is leading a lot of work in this space. So we're looking at not only at identifying best management practices, but also breeding alfalfa specifically for intercropping in these kinds of systems. And then we can look at more complex management that involves both overlap of different crops in time and space. So we're thinking about relay intercropping, where two crops might be grown simultaneously for part of the season and in monoculture for another part. An example of this is cover crop interseeding with cash crops. So we're starting to breed red clover for performance under the corn canopy. So we want clover varieties that can survive under these intense competition situations and then take off after the corn is harvested and produce good biomass for the next season. And finally, we're thinking about how to minimize tillage through breeding and variety selection. So as Matt Ryan mentioned earlier, there's a new dry bean project looking at dry bean production for the Northeast and in upper Midwest for organic production systems. And we're starting in 2023, we'll be evaluating dry bean varieties, both for performance in organic production systems using typical tillage management, but also evaluating varieties for no-till systems and uncovering the best varieties to use in these systems with rolled rye. So I think I'll leave it there, acknowledging the many collaborators on the projects that I've mentioned and the funders that have supported this work. And yeah, looking forward to the rest of the day. Thanks.
Hello, and I am an assistant professor of digital agronomy at Cornell University. I have a background of precision agriculture, conducting soil and crop surveys to generate maps. And today I would like to discuss how we can support New York State farmers in their experimentation towards soil health with digital agronomy. We know that farmers experiment on their farm. They use different techniques such as side-by-side, or it could be a strip, or sometimes they just compare two fields, adjacent fields. And their motivations are that they are trying to adapt tools or practices to their local farm conditions. They could also want to experiment to transition to a new practice, or introducing a new crop in their rotations, or adopt a new product, or sometimes it's to stop using a practice, for instance, in no-till. This farmer-led on-farm experimentation process is an indigenous process that's been used for thousands of years by farmers to evolve and improve their way of conducting agriculture. And if we look at those different motivations here, we can see that all of those are conducive to the transition towards sustainability. Because if we want farmers to improve the soil health over the coming decades, we want them to experiment with those new practices that we know are good for soil health. But why should scientists get involved in this endogenous experimentation process? I conducted interviews with 10 farmers in the state of New York last winter, and we asked several questions to understand first if they were experimenting, and also how they were experimenting and their motivations, such as what I showed you in the previous slide. And I was surprised by the answer from one farmer. So I asked the question. The farmer told me that they were conducting no-till on their farm, and I asked the farmer how long did it take to transition to no-till, and the answer was we are still transitioning after 17 years. So my take on this is that this is a quite slow process when you think about it, and all the practices that we can imagine today for transitioning towards soil health practices, 17 years seems to be quite slow if we want to make a rapid change and kind of reverse the negative impacts of agriculture on soil. And this is due to the fact that endogenous experimentation, so what farmers are conducting on their farm, is an iterative process. Okay, so in conventional research, we would replicate trials in time and in space, and with this, we are able to use analytics and draw conclusions. Farmers are more proceeding into an iterative process where they will change the protocol until they have something that works. So in an ideal conditions, it would look something like that, but in reality, it looks a little bit more like something like this here. And here we see like you try one protocol, then you improve, and then you come back to an earlier versions of the protocol, and then but you see that this process looks a little bit more like trial and error because there is no systematic progress such as what we are used to in conventional research. So the idea here is to try to support this process, the process that you see here, to help them accelerate that transition towards sustainable crop production. So what I suggest in this presentation here is to invent a new way to pull together this endogenous experimentation process that we know happens on most, if not 100% of the farms, with the new digital agronomy tools that we are used to work with in research, in conventional research. So how does this toolbox look like? First, when farmers experiment, they will always have some sort of observation. Sometimes they are looking at total yield, or they are looking at the weed pressure, or they are looking at the overwinter survival, and things like that they are looking most of the time visually with very few data records. But with digital agriculture, agronomy, we can contextualize those observations and therefore account for external factors. Okay, so there's a lot of layers of data that we can put on top of their experimentation to better contextualize what they have been working on. Second, we can use the data from multiple farms and this would generate a much larger database to work with and somehow replicate the experimentation that is implemented by farmers. And the third benefit of using digital agronomy toolbox is that we can use adapted analytics. Of course, if we're not conducting diligent experimental protocol, we cannot use techniques such as ANOVA, but with artificial intelligence, today we can work with diversified data sets to extract trends and better understand our world. You can imagine if we are working with data coming from multiple farms, how great of a potential this could be for advancing cover cropping. So for instance, for cover crops, there are so many variables that farmers need to figure out: the seeding rate, variety, termination dates, residues management, and all those. It would be very difficult for just one entity, one research program to figure them all out, plus their interactions. But if we work in a decentralized research, then we can use data from all those farms, centralize this information, and now look at the trends And what the trends that we see now, we can use conventional research to go and try to understand them better and to understand the causality behind those trends that we are observing on a larger scale. I proposed a project at the USDA Federal Capacity Fund in 2021, and this project was funded. So we are trying right now, what my research program is doing is to try to understand what are the processes of on-farm experimentation in the state of New York, and try to see like how we can contribute to this process. There is some literature, a new paper that was published in January 2022. If you are interested, you can scan the code here. And I want to thank you very much for this opportunity, and I will be happy to answer questions. Thank you.